Thank you to Dr. Martha Morell for being here today. She's Chief Medical Officer at NeuroPace and also Clinical Professor of Neurology at Stanford, and she'll be talking to us about trials for devices.
I was going to talk about medical device trials and in that context, medical devices, and really to highlight some of the challenges and the differences from trials of anti-epileptic medications. So I'll begin by a disclosure. So I work at Stanford, and I have a second job also as the Chief Medical Officer at NeuroPace, which is a company that has developed a medical device for treating epilepsy. So just to begin with, of course, we have devices that are approved by FDA, and the first being the vagus nerve stimulator. And this was approved after 10 years of clinical trials, of clinical development. So a long time and a lot of work. You are, I'm sure, well aware that the VNS provides open loop or scheduled stimulation, with a new version providing closed loop or responsive stimulation when a heart rate that exceeds a predetermined level is detected. So the other device that is not available in the United States, it's not approved by FDA but is available outside of the US, is another open loop device, the Medtronic Activa, which takes the basic premise of treatment of movement disorders, Parkinson's, with scheduled thalamic stimulation, and picking a different target, anterior nucleus of the thalamus, provides scheduled stimulation for epilepsy. The third device is the responsive neurostimulator, brain responsive neurostimulator, the RNS system. And this was approved after 15 years of development, technical and clinical development, for treatment of medically refractory partial onset seizures in adults. And the basic premise here is similar to cardiac devices that sense and stimulate. So in this case, the neurostimulator is continuously sensing the electrocardiogram, and when specific patterns previously identified by the physician are detected, it delivers stimulation response. And the stimulation is also determined by the physician. The physician is able to review data stored by the neurostimulator and uses that in addition to patients' clinical reports to assess the adequacy of response. So that's what we've got, or what we're soon to get. And the point I want to make today is that it's really difficult to get devices approved. And there are misconceptions, and one of the misconceptions is that it's harder to do a drug trial. The other misconception is that for devices you only need one study, whereas with drug trials you need more than one. There is a misconception that you don't need to have controls in device trials, and that there's less of a placebo response, that it doesn't take as long, and that it's less expensive. So I'm going to explode those misconceptions. To begin with, though, and this is not anything I was aware of until I started getting involved in the NeuroPace project, and that is that within FDA there are different agencies that regulate medications, and therefore anti-epileptic medications then regulate devices. So for medications, it's the Center for Drug Evaluation and Research, but for devices, it's the Center for Devices and Radiological Health. These are in completely different buildings. They are completely different people, and they generally do not speak to each other. So the level of expertise concerning epilepsy may be very different across the two groups, and in fact generally is, because the Center for Drug Evaluation and Research has had many more epilepsy products to evaluate and to determine whether they're going to approve or not than the Center for Devices in Radiological Health, which until recently had only one device it had ever considered, and that was VNS, and as I mentioned, that was 1997. So it's a whole different group of individuals who may have very different ideas or level of expertise about epilepsy and different ideas about what is required for a therapy to prove that it is worthwhile. So the other point within FDA is that within CDRH, they classify devices into 3 different categories. And Class 1 are the simple devices, and that includes things like tongue depressors and blood pressure cuffs, which are considered medical devices and are regulated by FDA. And Class 2 devices are those that have some type of performance standard, but it's not anticipated that there's any risk of harm to the patient. So those types of devices would be things like syringes and wheelchairs. So I guess if there was some gross malfunction, there is some possibility that that a patient might be injured, but it's just really not within the expectation. And then Class 3 devices, which is where all the epilepsy therapies have fallen, are those that are important. They may not be, their performance may not be essential to life, like a cardiac defibrillator is considered. So any malfunction would be considered to have a likelihood of causing death, but they are important to support life and sustain normal quality of life, and the malfunction of them could cause injury. So this is where these fall, and these are under a much more stringent scrutiny. Also within devices, there's this strange thing that is historical, that before, if a device was approved by FDA before the 1970s, it is considered to have predicates. So before the 1970s, there wasn't much required to approve a device, and so all the devices approved before that time were grandfathered in. And if you can show that you are similar to a device approved before that time, then it is considered that you have a predicate. So for example, a cardiac pacemaker may claim as a predicate some of the early devices approved, and then if there's a predicate, you just have to show that it's substantially equivalent. If it is, then you don't have to do a study. You just have to show some evidence that it's technically going to work, and maybe have to show a little safety data. If it's not substantially equivalent to a 510K product, then you have to go down a different pathway where you have to provide a little bit of data, but not a whole lot. If there isn't a predicate device, which is the case with all of the epilepsy devices, then you have to do a pre-market approval application, and that is a whole different animal. And it is a big trial and a big commitment on the part of a company. So that would be where VNS, DBS for epilepsy, and the RNS system fell. The requirements for a pre-market device are at least 2 trials. And the first, it's not phase 1, phase 2, phase 3 like it is with drugs. It falls into 2 categories: feasibility, and that study is designed to show that it is safe and that there is some reason to believe that it is effective. So it's not an efficacy study, but if there is no efficacy demonstrated, then it is unlikely that will allow it to move on to the next stage, which is a pivotal study. And the pivotal study is powered for efficacy as well as safety. But another difference between CDRH and the drug group, Seizure, is that CDRH will not allow you to use the word efficacy. You have to use the word effectiveness. And this sometimes creates a little confusion because in the drug world, effectiveness is considered to be a combination of efficacy and safety. But in the drug world, they're very, very, very sticky about that you are evaluating safety and effectiveness. So the post-market trials are supposed to show safety and not just acute effectiveness, but also long-term. So the requirements for following patients with a device are much different than how long you need to follow patients with a drug. The outcome studies for devices are intended or required to be hypothesis-driven. You need to demonstrate that the subjects are typical of the target population. You have to randomize and blind if there is any way that you can do so. They also require that you use comparative study designs and that you have outcomes that are objective and can be validated as real outcomes. So that's probably not so different than with drug trials. But how can you use controls in comparative device studies because you're doing a procedure? Well, they say that you can compare treatment to no treatment if you already have objective measures of effectiveness and you can demonstrate that there's really no placebo effect. But you need to use a placebo if you're going to compare to an ineffective device. So that would be a device that is programmed to no treatment or ineffective treatment, which is how the VNS trials were designed. And that was that every patient was implanted with VNS, but half were treated with a stimulation that was felt to probably not be effective, and half were treated with an effective level. Or you need to use you can use an active treatment if it is considered that withholding treatment is not ethical. So that really falls more into the devices that are considered to be life-sustaining. And then finally, they may consider historical control, and there has been a lot of discussion of that with device trials because all patients are exposed to a procedure. Is it appropriate, since there is so much data on the natural history of epilepsy, to simply control the simply compare the response to a device to what we understand to be the natural history? And typically, CDRH has said no, although we all keep trying. The regulatory standard for approval for safety is that you have to show that there's reasonable assurance that the benefits with the intended use outweigh the risks. And you need to show valid scientific evidence, and that has to include animal studies, human subjects, and also non-clinical investigations, which are known as bench testing. So you have to show that the device will consistently perform as expected under extreme conditions like high temperature, like 50-foot drops, like having somebody stamp on it and such. And then for effectiveness, you also need to show that the use of the device will provide clinically significant results. And for that, they look at well-controlled investigations. So the safety, you got to show animals, humans, and these extreme tests. And for safety, for effectiveness, it's all the clinical trials. And then they evaluate the probable benefits to health against the risk of injury. And they will look at the diversity in the patient population. They also look at user experience. So this is different than drugs. They pay very close attention to whether trained users have a different patient outcome than non-trained users, and also level of experience. So you need to show that somebody who has treated 14 patients with the device doesn't have a different patient outcome than somebody who has treated only 1 or 2. And that is a big responsibility of the the company that's developing the device to provide them data based on training and physician experience. And then, of course, they're going to look at whether there are other options available that are already approved or cleared. If they feel that your device has any inferiority to something that is already approved, then they won't approve it. So it is much less likely that you will get a Me Too device than it is that you will get a Me Too drug. So what are the challenges? Well, it may be hard to develop a comparative trial because everybody has to be implanted. And when patients are provided stimulation, it is possible that there will be a perception. So it's harder for some of these devices to really achieve a blinded study. And then, as I mentioned, the user skill may be a variable that's difficult to control. And then also, when you do a device trial, you are committing to a study that is going to go on probably a decade. That's the general requirement now. The other problem, which I think a lot of people don't understand, is that technology changes very rapidly. So with our iPhone, we're getting updates pushed to us every couple of months. But with a device, once you have the device, you can't change it without undergoing a great deal of scrutiny from FDA. So you may end up with technology that is not exactly what you wished it would be a few years down the road, but you can't change it. And then also, it's really hard to know the dose when you start the trial, particularly if it's a device that has never been used at all, like with the RNS system, or has never been used for that indication, such as with the DBS. So just to have time to have questions, I'll go over this very quickly. But the challenge here is that you really don't have a true placebo because you have a procedure. So there is going to be an effect of the procedure, and some of this may be the patient's expectation of benefit or the patient's wish for benefit, but the other is a real effect of the mechanical effects of the procedure. And I'll show you how this definitely was the case with the brain stimulation devices. And then you've got an issue where you have the physician managing a device. So it is possible that it's going to be pretty easy for the patient and the physician to know whether their device settings are being changed. So that's a challenge to maintain that. And then one thing that really VNS ran into, which was, I think, kind of unfair, is because patients can feel stimulation of the VNS, FDA decided that they needed to do a stimulation current that they felt would probably be effective, but that had to be compared to a stimulation current that the patient felt, but was less than what Cyberonics guessed or guesstimated would be effective. But it is very possible that that low current did have an effect, and therefore that would make it more difficult to demonstrate a difference between the low and the high. So the effectiveness may be harder to establish when you use the standard statistical methods for demonstrating effectiveness that FDA requires. And then I mentioned the human factors. And the training becomes a big issue in ongoing training. What does that mean for a device company? Well, it means that, and this is so with cardiac devices too, that you are involved with the physician user at the beginning and maybe forever with the device. So with devices, companies provide field clinical engineers, or they may have a different name, who are there when patients are implanted, who are there when patients are seen, and who are engaged in ongoing training with the physician. What about adverse events? Well, you need to consider which adverse events are related to the surgical procedure, which are related to the device itself, like with VNS, the effects of stimulation. And it's important to differentiate those because it's also important to counsel patients about which adverse events are procedural versus treatment related. And then the other challenge is that you need to monitor adverse events for a very long time. And if there are adverse events related to stimulation, some of those may be acute, but there is a very reasonable concern on the part of FDA that it may be that if you stimulate over a very long time, have adverse events that are related to providing chronic stimulation to whatever the target is, whether it's the vagus nerve stimulation, vagus nerve or an area of the brain. And then you also need to, all of these have batteries, and the batteries run out, and the device has to be replaced. And so you have to track adverse events that may be related to the replacement of the device or related to explantation of the device, such as adverse events from trying to remove a lead that may be adhered to the vagus nerve or may have gliosis in the area of brain in which the lead is implanted. So what are we really here? Well, device development is ongoing and iterative because you continue to understand what the user needs, and technology will continue to be improved, and that will include the materials that are used for the inside and outside of the device, the hardware which has to do with integrated circuits, software which is more changeable than the hardware, and also determines the user interface or ease of use, and then batteries. The batteries are not made by the medical device companies almost always. The batteries are made by battery companies, medical battery companies, and so the longevity and the size of the battery are very important to device performance. And so you also may be in a situation where you're doing a study, but you have improved technology and your study isn't finished yet. But you have to use the old technology in your study because FDA will not allow you to change the device that you are using in the midst of a trial. Then when you get the new technology, FDA may say, well, this is not the same device, and therefore you have to do additional studies to prove that this device does not perform differently. And so I'll give you one example with the RNS system, which now has 2 leads. In order to introduce a 4-lead system down the road, FDA has indicated that may be required to do a new controlled, double-blind, randomized controlled trial to show that 4 leads is not different than 2 leads, and that's a major commitment. So what's the issue with designing these trials? And I'm just going to go straight to the points here. One of them is, do you require many seizures a month or fewer? So if you have many seizures a month, it may be easier. You may need a shorter baseline. You may need a shorter blinded period. But you're also going to restrict the numbers of patients who are eligible for your trial. Another consideration for device trials is, do you exclude anybody with risks, more surgical risks, such as patients who are anticoagulated, patients who are older, patients who have any significant medical illness, and do you include those with psychiatric comorbid conditions? And this actually, particularly with DBS and with the RNS system, there was a concern that stimulating in certain areas of the brain might exacerbate depression or anxiety. And also with RNS system, that individuals who were paranoid or had any well, really largely paranoia would be concerned that having such a device could be capable of controlling thoughts or broadcasting thoughts to others. So there's all of these issues. And then do you, since devices involve a procedure, it's not a big operation, but it's a procedure, do you only let people in device studies that are absolutely not candidates for traditional epilepsy surgeries? And this came up with the RNS system trials, and it may have come up with the DBS trials as well. I don't know that. And that was a discussion among physicians about whether it was ethical to allow patients to choose to be in the RNS system trial if they were a candidate for a epilepsy resective surgery, even if the patient had said, I don't want a resective surgery. And ultimately, the decision was that the patient's choice could direct their path. Now, the blinding, the patients are randomized to active versus sham. And I already touched on the issue with VNS, and this has also been an issue with trigeminal nerve stimulation, because patients most definitely feel when the trigeminal nerve is stimulated. It turned out not to be an issue for the deep brain stimulation and RNS system trials, because it turns out that patients have no perception that they're being stimulated. So that was easy. But it was also required, in order to maintain the blind, that there be 2 investigator teams at each center, one that managed the device, and so they obviously knew where the patient was randomized, and the other group that was involved in collecting the efficacy endpoints. So this group counted the seizures but had no interaction with the device. And that system worked for both the DBS and the RNS system trials. The blinds were maintained for the patient and for the physician. But there are observations about trial design. So with medications, there are a number of ways of looking at it, and one of the ways is crossover designs. But it's become apparent that for neuromodulation that there's a carryover effect. So even if you stop providing stimulation, the effects of stimulation persist for some period of time. So devices don't have the option of doing crossover. So there has to be 2-arm studies, a treatment and a no treatment group. But the issue with that is that there's an implant effect, and there's also an effect of stimulation that emerges over time. So this has become a challenge because we cannot use the same length of study that are used for drugs, and everybody found that out the hard way. Also, for device trials, you change the stimulation settings. You don't do that in drug trials. You randomize people to a fixed dose. In trials of devices, you are able and you must change stimulation. So the optimal dose of stimulation for a device is often not known when the device is approved. Let me show you the RNS system trial just to show you where the problems were. So we designed it based on drug trials because what did we know? We had no idea. So there was a 3-month baseline after which the patients were implanted, and then there was 4 weeks where people just got past the surgery, and then on week 4 they were randomized to receive stimulation or to continue to or to get a sham stimulation. The first 4 weeks, the stimulation was adjusted in the active group, and then post-op weeks 8 to 20 were used for the blinded evaluation period. That was a 12-week period, and then everybody went into an open-label period where they were all treated. So let me go in the right direction. What was the issue here? Well, the issue was that there was an implant effect. And when patients were implanted, everybody, even before they were randomized, had a 25% reduction in seizures. And then during months 1, 2, and 3 of the blinded period, which were months 3, 4, 5 after implant, there was a progressive differentiation between the sham group and the treatment group. So what we learned from this, the hard way, is that the implant effect lasts 5 months, and that it may be difficult to show a difference in response between those treated and not treated unless you look for long enough. And fortunately, in this study, we did look long enough, and there was a statistically significant difference. But if we hadn't waited that first month, or if we had randomized people right at implant, it is very possible that it would have not been possible to show that there was a difference because the implant effect would have obscured that. What's the other phenomenon? Well, the other phenomenon which has been observed with VNS, DBS, and the RNS system experience, and this is RNS system data, is that people continue to get better with time. And this slide demonstrates that at year 2, year 3, year 4, and really extending to year 7, we see continued improvement. And to some extent, this is because stimulation settings are being adjusted in ways that may be more beneficial. But I think everybody now believes, everybody who's been involved in these trials now believes that even if you left everything the same, patients would continue to get better. So this suggests that you will not be able to appreciate the full extent of the response if you do a design like a drug trial, where most patients are followed only 6 months and in some instances a year, but really not longer than that. FDA does require usually a decade of follow-up, and that may seem burdensome, but it turns out for neuromodulation devices it actually is probably necessary. What are the pros, or what are the issues? Well, one issue may be that we need to lengthen our randomized blinded period longer than what we do with drugs. And the pros are that gives you time to really get back to your baseline so you get past whatever effect is exerted by the procedure. And then you also can see this emergent effect. But the problem is are patients going to be willing to do this? These are ill people. They may find it very difficult to commit to a trial where they're not going to be treated for a long period of time, and it may not be ethical. We know that people die when they have bad epilepsy, and that SUDEP rates are high in this population, and it may not be ethical to make people be in a placebo arm for 6 months or 9 months or even 12 months. And then it becomes so expensive to do these trials that it may not be possible at the end to ever recoup the investment that's made. So we may find if we do trials that go on too long that companies just aren't able to do it. So then another question is, do we go to the open label data? Because we really can then see these changes over time. There's an argument that if you use open label data that you're simply seeing regression to the mean. But I think most people feel that when you see improvements at 5, 6, 7 years, consistent improvements in the large population, that it is unlikely that is a regression to the mean, but it's always a possibility. So there's a lot of interest in whether we could use biomarkers. And the pros of this is that you would use the clinical data, of course, but you might be able to supplement that with objective measures. And this is an area of interest with the RNS system because it gathers data from the electrocorticograms. The interest is whether electrographic features like electrographic seizures or numbers of spikes or overall power or spectral frequency analyses could be objective measures, and you could use those to demonstrate favorable changes that would be less prone to any bias because of patient perception. But of course, we don't know yet whether these are correlated, and that work obviously needs to be done. Statistical considerations for device trials are different. Drug trials enroll thousands of patients, but for devices you enroll hundreds. So it is difficult when you have a disorder like epilepsy where the events that you're measuring are highly variable across subjects. And so the analogy would be trials of antihypertensives, where you're getting multiple measures within an individual, like you're getting multiple days of whether people have seizures or don't have seizures, and also there's just a wide dispersion. In those situations, parametric statistics are really considered for these other disease states to not be appropriate because you get non-normal data. And if you use parametric statistics, you may not be able to show true differences. So here's an example from the RNS system trial. So this is the baseline seizure frequency for every 28 days in patients who enrolled in the pivotal study. And you'll see that it's definitely not a normal distribution. And if you looked at the median, you certainly do not get a sense of the range of the seizure experience, nor does the mean provide that expression. So in these instances, you need to use different analytic techniques than have been used traditionally in epilepsy trials. And then to kind of finish up on what other differences are. Drug companies are not required to do post-market or post-approval studies. Sometimes they'll do so, but in the past, physicians have looked at these dubiously because the thought has been these have been marketing studies, and there's been a lot of discussion, including pros and cons in the journal Neurology, about whether results from post-market studies should be considered as valid. For devices, FDA requires these studies. They are real scientific studies. They want to gather additional open-label data on efficacy and safety in a patient population that may be more diverse than those who were enrolled in the pre-market or pre-approval studies. They also want to continue to get data on the effect of the surgeon training on safety largely, and on the physician who is programming it, training on the patient outcome. Now the cons are, of course, that patients may not want to enroll in a trial when they can get the device without doing so. And you don't necessarily have a prospective because patients aren't willing to do that. And of course you aren't randomizing patients, everybody's being treated. To try to get a baseline or to do a blinded period, you really run into even more problems about whether this is unethical, because you have a device that you know to be effective, and not only do patients not want to enroll in a baseline period, you are withholding an effective treatment, and that becomes a difficult argument to make. Another way to gather this post-approval study is through registries, and this is physician reports of how patients are doing. This is probably somewhat helpful in understanding user experience and effectiveness, and it does allow you to get data on very rare adverse events. So that might be a device malfunction or something, well, I'll give you an example. There was a cosmic event in Seattle. There was literally a cosmic event in Seattle, and one RNS system, neurostimulator, briefly turned itself off because it is built to do that when there are more cosmic rays. I'm not kidding. So this is something that, you know, is obviously rare, and it gives you some information about that. But it also addresses the requirement that patients be tracked. So another difference with devices and drugs is that you own the patient when your device is implanted in it. You know who the patient is, you know where they live, and you are supposed to track that device until it is explanted or until the patient expires. So this addresses that requirement. But the cons is that if you aren't getting 100% ascertainment, you may be getting a very selected data set. And it also may be difficult to get patients and physicians committed to do this collection, so you get partial reports, or you'll have certain physicians who do and other physicians who don't, and so it may be a highly selected population. And then you also have data that, you know, how is the patient doing? Well, it's a retrospective baseline that may work for you or against you. And then finally, in neurology, there are not systems in place to support registries. So the American College of Cardiology has registries similar to RAAN. That professional organization has registries, and the members of the ACC register all of their patients implanted with devices in the ACC registry. This has been in place for several decades, and we don't have that kind of a system that is not company specific, and that is really what would be helpful to make registries powerful. So to close, how do I sum up the differences? Well, with a drug, there's not really a change in technology. Drug is drug. It's, you know, not going to change. Maybe you'll get an extended release. Maybe you'll get a different dosage form, but basically a drug is a drug. But with a device, there are constant iterations with advancements in technology. With a drug, how a physician uses it is largely not influential on how it works. Now, of course, physicians have expertise in terms of how quickly to start it, so that may influence patient tolerability, or may have more knowledge about drug-drug interactions. But the MD technique is very influential on the response to devices. The ability to adapt treatment with use for drugs, and certainly in trials, is low. You can't switch a patient to a different dose arm in a drug trial. You can in Europe, but you can't in the US. FDA won't accept that. But you certainly can with devices, and in fact, you should and you must. And then finally, with a drug, the patient is anonymous as far as the pharmaceutical company and the FDA is concerned. But the patient, it must be tracked with a device. And so that does allow devices to continue to gather very helpful information over the entirety of that patient's experience with the device. So these are the big changes, and I hope I've made the points that anybody who gets a device to market for epilepsy deserves a lot of credit, because it's very difficult and it's incredibly expensive. We do need 3 trials to get a device approved, at least a feasibility, pivotal, and post-market. You do need controls, but it may be difficult to figure out what your placebo is. The commitment to investigation is many years long, so that adds to the expense. And you also have the issue that you are, your technology is kind of chasing the trial. I mentioned they're very expensive because of the cost of the device, and because there's procedures, and because of all the patient support that's provided. It costs just as much money to get a device approved as it does a drug. But as the recent Medtronic DBS for epilepsy and the RNS system trials have demonstrated, it can be done. But it's so hard that there isn't the flood of enthusiasm for bringing new devices to market as there is for anti-epileptic medications, which I think is a shame, because I believe that neuromodulation is a new treatment paradigm that is going to profoundly contribute to the treatment armamentarium that we have. So that's it, and I would love to hear if there are any questions. I see that somebody does not have a microphone, so there is a chat box, I believe. Kara, is that correct, that people can ask questions?
Yes, people can ask questions through the chat, or if they do have a microphone, can ask them verbally as well.
I have a question for Dr. Morel.
Please.
Sorry, I'm not sure if that's directly relevant, but the Sente trial, or the DBS, what's the truth about the approval? Why was it approved when the efficacy was, to me, was not very different from the RNS as far as the outcome of the study?
Well, what FDA has publicly stated is that they're concerned that the trial did not meet its effectiveness endpoint, and the reason for that is that a patient was excluded, and when that patient was included, the trial did not meet the statistical endpoint. There was a second patient who had been excluded during the trial because the feeling was that the patient was having non-epileptic seizures, and then when the analysis was done, that patient was included again, and if that patient was not included, the trial did not meet its efficacy endpoint. So this is what has, what is public record from FDA. So they felt that there were 2 manipulations that had to be done in order for it to meet its efficacy endpoint, and if you included the entire intent to treat population, it did not. The second was that FDA was concerned that there was a difference in adverse events between the treated and not treated group, and that there was a statistically significant increase in the frequency of adverse events related to memory and concentration and depression in the group treated compared to not treated. So they felt what FDA decided was that the benefits did not outweigh the risks. Having said that, I know that FDA has reconsidered that decision, and my personal expectation, so this is my personal expectation, I have no inside information, is that ultimately it will be approved because the study has continued to gather long-term data. Like the RNS system, they have data as long as 11 years on these patients, and they continue to provide that to FDA.
So for a device, the device can be approved based on like post-marketing studies or data, even if the pivotal trial may deem not meeting the outcome. Is that accurate? Or they would require another study in order to approve it? In general.
You know, a lot of this, and this was a surprise to me, a lot of this comes down to the individuals who are on your review group. So the way that it works in CDRH, and I think Cedar as well, is that there is a review group which is relatively small, and you have one reviewer who is an engineer or a technical person, and you have another reviewer who is an expert in manufacturing and quality systems and so on, and then you have a statistician and you have a clinical reviewer. May only be one clinical reviewer, and so it's really that individual who goes to the larger group and says whether they believe that the device is sufficiently effective and safe. So there is a lot of conversation with this review group. You get to know them extremely well, and depending on whether you can convince the clinical reviewer, for example, you can convince them that removing a patient and then adding back another patient was legitimate to meet statistical significance. If you can convince them that really there aren't long-term problems with memory and depression, then that clinical reviewer may change his or her mind. So yes, and you may continue to plead your case. They may also change their mind based on your post-market data. So if there is all of a sudden adverse events that emerge at 6 or 7 years, for example, in your post-market study, you don't show that the effectiveness in the post-market study is as good as it was in your controlled trial, they can pull your approval. And they do, and they can rescind it at any time. So the conversation with them does not stop after you are approved.
Thank you.
So there's a question, what about new indications for previously approved devices? Well, I think one good example of that would be the indication for depression with the vagus nerve stimulator. So once you have a device that there is experience as far as the performance of the device, it's not going to malfunction, the procedure to implant, like with VNS showing that it's acceptably safe, then you can look at new indications. And those trials will be designed to demonstrate efficacy. Of course, you will continue to gather information for safety. You may not necessarily need to do a feasibility study, although probably you will have to, because FDA talks to you about what your effect size needs to be. So they'll say, well, you have to have an improvement for depression and, for example, the Hamilton score of this many points for us to consider it effective. But here's the other challenge that I think we have to appreciate. So FDA did approve VNS for depression. They decided they made their endpoints, the benefits outweighed the risk. But then Center for Medicare Services, Medicare, decided that the effectiveness data did not show enough improvement. So CMS declined to pay for it. And when CMS made that decision, all the private insurers said, well, we're not going to pay for it either. The private insurers generally, not always, but generally will do what Medicare decided. So that completely killed VNS for depression, because although it was approved, nobody could get paid for it. So it's a combination of FDA and what the insurers decide that decides whether a treatment is going to be available. As far as whether there are going to be trials, and this may have been your question, for new indications for DBS or for the RNS system, I think the answer is going to be certainly there will be. Although I am not aware of any going on with DBS right now, and certainly there have not been new trials launched with the RNS system. With one exception, there was a trial of deep brain stimulation for depression. The anatomic target was the anterior limb of the internal capsule and nucleus accumbens. That was a negative trial, and as of this time, there are not any other DBS trials for indications beyond movement disorders and epilepsy outside of the U.S. And any other questions? Well, I hope this has been helpful to you, and I hope that I've told you something you didn't know before, and I hope you have a healthy respect for Cyberonics and for Medtronic and for NeuroPace for successfully completing these efforts.
Thank you very much.
Thanks, everybody.